Namah Shivaya ma'am, my name is Tanmay and my project is DNA motif recognition using hybrid genetic algorithm and particle swarm optimization algorithms. The contents are the abstracts, introduction, project objectives, methods, current status, summary, references, and background is also involved. So for the abstract, we are using motif recognition to understand the gene regulations. This project uses a hybrid between genetic algorithm and particle swarm optimization methods to identify common patterns in DNA sequences. This model uses the GA and AI. It enhances the performance using the particle swarm optimization. Introduction for is DNA short motifs are short recurring patterns in DNA sequences. They are crucial in indicating our transcription factors sites. Important of discovery, it helps in understanding gene regulation methods. It's used in many. It has many applications such as bioinformatics, genomics, and drug discovery. Project objective. The project objective is to create a hybrid between GA and PSO for motif detection. These are used for more efficient detection compared to other primary methods, and it evaluation performance is on the DNA datasets. So genetic algorithm, it uses the principle of natural selection and gene evolution. It takes the it checks the fitness for each individual, and after checking the fitness for each individual, it moves over to the selection of the fittest individual. After creating the fittest individuals, it applies crossover to create a mutation for a it crossovers the fittest individuals and creates a mutation to create a new population. After this, it repeats this until it converges. So the particle swarm optimization is inspired by the flocking behavior of birds. The idea of the particle swarm optimization is that a group of particles move through the solution space influenced by their best and global positions. Yeah, particle swarm optimization uses the position and velocity updates to check for the next optimization. It randomizes the position and velocities. Next, it checks the each particle's fitness. After checking the fitness, it updates the velocity and position based on the personal and global best positions, and it repeats this until convergence. So for the question of why hybrid genetic algorithm plus position swarm optimization, it is to leverage the strengths of both approach. Genetic algorithm ensures the diversity and PSO improves the convergence. It GA adds randomness and diversity to prevent premature convergence, while as PSO refines the solution more efficiently. So, for this purpose, we create a hybrid of GA and PSO as these complement each other somewhat. So, for the how much process is for the methods which are using, first is pre-processing and encoding. We collect the DNA sequences and encode sequences using binary or one hot encoding. Next, we initialize with a GA method. We create the initial population of motifs and use evaluate using the fitness function, where we create where we take the fittest model and mutate it to create the next population, and we to converge the answer. Then we use the PSO to integrate with the GA. It refines the motif by converging the swarm. So for the workflow, we first initialize the sequence patterns. After initializing the sequence patterns, we evaluate the fitness. But after evaluation of the fitness, we do the GA operations. After the GA operations, we go to the PSO updates. After updating the GEO operations with respect to the PSO, we replace the methods by evaluating the fitness again. After replacing the individuals, we iterate again. We again initialize the new individuals and then go through the same process. We repeat this a few times until we get the output. We combine both algorithms in an iterative optimization loop. As for the implementation progress, for now we have completed the preprocessing and encoding part. I have also given the written the GA methods and PSO methods on with which will work separately. Uh, as for the code part, this is the code. If we're importing OS, which is used to handle file paths, then NumPy, which will arrange it as an array or matrices. Random is for randomization, and glob is used to search for .gaspr files. Then the file path is given here, and we check make the file. So we create a method that is called parse.js per pfm. What it does, it creates a file which stores the PFM matrix, which means position frequency matrix. These PFMs are converted into numpys and are stored corresponding to all the ACGT, and these are used to determine the common Euclid patterns. Then we have the another method which is called PFM2 consensus. What it does is it creates a sequence where DNA sequence where it is most likely based on the PFM. For each position in the motif, it picks the base with the highest count in the column. Then we have the method one hot encoding. What it does is it converts each DNA sequence into a one hot vector. It maps each base to a 4-length binary array. So for each and every sequence, it creates an array. This format is ideal for matrix based motif alignment. Next we have the GA process. Here it initializes the population, which is all the motifs we have by creating the one hot encode method. Then we use the fitness function. It checks the how well motif is how well the proposed motif is across all sequences. It it builds a profile position matrix based on the segment and scores how strong the segment is. The higher the score, the better the motif is, and based on the motif, it selects the highest population, highest numbered motifs, and then it crossovers the parents. After crossing over the highest selected individual as a parent, it mutates and creates a new individual so that we can have a diff. It mutates so that it can create new population to repeat the convergence. Then we have the particle method. So for the particle swap method, it first initializes a position, the velocity, the sequence length and the motif length. Then it creates the best position and the best score. Best position, it considers the current position. Then it updates all the best positions velocities with respect to the other way. It uses the given formulas to check for the updated velocity, and if the updated velocity is better than the previous velocity, which is the current score, then it replaces the current particles. So after it replaces the current score, it goes to the same process and it converges. Uh, So as for the remaining topics, I have still yet to implement where GA and PSO. I need to implement them both combinedly. Then I need to check for the errors and see if I need to correct any process. And if the desired output is not desirable, I need to optimize it better to get a better output. So for the summary, I have presented a hybrid approach for the GNPSO using motif recognition. I have combined the strength of both algorithms for better results. The implementation and early result for each of them separately are promising. Next, I need to tune them, etc. As for the references, these are the references where I have taken.